First, let's talk about the background information. Magnetic resonance imaging is a medical imaging technique which utilizes the fact that human bodies are composed of cells containing hydrogen ions. The strong magnetic field induced by the MRI scanner can cause the positively charged hydrogen ions to spin in an identical manner. The direction of the spin of the protons varies with the strength and direction of the magnetic field. The removal of the magnetic field lets the protons return to their original state by precession. We can evaluate different tissue types by the varying precession rates. It is commonly used for imaging of brain because it is non-invasive and does not release ionizing radiation. It also has the advantage of showing superior soft tissue contrast. We need to reconstruct the data of MRI images because the data is initially collected in K-space, which is an array of numbers representing spatial frequencies. We need to convert the K-space data back into an image. Artifacts in MRI are generated by head movements and metal implants. They cause distortion, thus affecting the resolution of the image. Furthermore, the accuracy of data reconstructed will be lower. This is why denoising work needs to be done. We're going to focus on brain images that shows the diffusion directions of the fluids in the brain, and then we are going to investigate the microstructural integrity of white matter in the brain. Here are the main objectives of the study. First, we are performing data reconstruction using diffusion tensor imaging. Second, we are going to generate a fractional anisotropy map to visualize diffusion directions. Third, we are going to process a diffusion-weighted image dataset. Lastly, we are going to perform denoising using a filter mask. Now, we are going to talk about the laws, theories, and definitions used in the data reconstruction part. Again, in this study, we are going to visualize the diffusion directions of the fluids in the brain. Diffusion is a spontaneous and random process. Particles drift down the concentration gradient from a high concentration area to a low concentration area. The directions can be detected by MRI. The MRI signals are sensitized to water diffusion. Studying diffusion of fluids in the brain aids studying tissue microstructures. The information that can be obtained include the permeability of membranes inside the brain, the density, and the compartment sizes of the tissues in the brain. The technique used to obtain the data is diffusion tensor imaging. It maps and characterizes diffusion of water molecules in brain rather than showing the anatomical structure. To calculate diffusion tensor, we first create a 3x3 tensor model, and then we can derive the fractional anisotropy map, which gives information about the degree of directionality and mean diffusivity. It is useful in detecting neurological disorders, injuries, and brain connectivity. Here are the equations for calculating the tensor model for each voxel. With signal intensity measured, p-value calculated with the equation below, and direction of gradient already known, we can evaluate the tensor matrix. A higher p-value means higher diffusion weighting. The duration for diffusion to occur at the point will be shorter. The diffusion matrix is in the form shown in the figure on the right. In the matrix, the diagonal elements are proportional to the diffusion displacement variances, and the off-diagonal elements are proportional to the correlations of displacements along these directions. With all the values calculated, we can visualize all the diffusion directions of the liquids in the brain. We can further derive the alternative representation of the matrix by eigen decomposition. When the eigenvalues are the same, the voxel is isotropic. Diffusion to all directions are constant. When one of the eigenvalue is larger than the rest, the voxel is anisotropic. There are different dimensions in different directions. In the study, we'll import this equation in MATLAB to evaluate the tensor. Another important unknown is the apparent diffusion coefficient. It quantifies the rate of diffusion and is calculated using signal intensities at 2 or more b values. In the study, we divide the tensor matrix by ADC, to evaluate the fractional anisotropy. Fractional anisotropy shows the direction and magnitude of diffusion, which is quantified to values between 0 and 1. The fractional anisotropy map is a grayscale that helps visualizing the values across the brain, with the brightness implying how well organized the white matter at that point is. With the diffusion directions visualized, brain abnormalities can be identified swiftly. Now, let's move on to the laws and theories of noise reduction. We use diffusion-weighted images to perform denoising. The image measures the random Brownian motion of water molecules within tissues. It measures the diffusion coefficients inside tissues, which lowers in areas highly packed with tissues and cellular swelling. The reason of choosing diffusion-weighted images is because it experiences high level of noises, especially with high B values. The signal of interest might be overwhelmed, and the bias will be generated during evaluation. The method we are going to use is a directional filtering of K-space. Using the tensor values collected from the data reconstruction part, we select one image corresponding to the B-log value and 30 images corresponding to a self-decided B1 value. K-space is filled with Cartesian filling approach with the equations listed below. The diffusion-weighted images are processed with quadratum Fourier transform and quadratum inverse Fourier transform. Fast Fourier transform and shift operations are involved. This helps reconstructing the images from the spatial domain to the frequency domain. It also helps centering the zero frequency domain component. In the process, noise is added before the quadratum Fourier transform, and the denoising method is applied between the quadratum Fourier transform and the quadratum inverse Fourier transform process. We start with adding white noises to the noise-free image set. I-noisy and IGT are nth noisy and noise-free images, while NRE and NIM are the noises added to the real and imaginary channels. We achieve denoising by applying a filter to the image set. The filter is a petal-like pattern. It is a mask aligned with each gradient angle. After applying the mask, the less directionally affected information should be lying outside of the pattern, so the values inside of the pattern is retained and the values outside are normalized to the mean K-space data. Now, let's move on to the experimental setup and results of the first part, data reconstruction. First and foremost, we obtain the data from Human Connectome project website. Human Connectome project is a research initiative aimed at mapping the neural connections within the human brain. It tries to create a connectome. It provides detailed and clear map of the brain's structural and functional connectivity. The data collected by the Human Connectome project is openly available. Then, we simplify the data structure by using the choose slice function to extract the image's second dimension. Next, we decide the diffusion weightings, which are actually the B values. A higher B value means higher diffusion weightings, which is optimal in this condition. The B value chosen is 995, and the corresponding images are saved. Next, we record the B vectors, which denote the diffusion directions. After that, we concatenate T2 weighted images with the data into a single matrix. The tensor data are visualized by the figure montage function and is shown below. Then, we apply a Vienna filter to perform simple noise reduction with the Vienna 2 function. The next step is to calculate the apparent diffusion coefficient with the formula introduced earlier. A series of operations, including division, symmetry, and eigendecompositions, are applied to the data. Eventually, we evaluate the fractional anisotropy. Here are the fractional anisotropy and the colored fractional anisotropy map generated. Now, we move on to the noise reduction part. First, we load the tensor matrix generated earlier. The total number of dimensions of directions is 30. After defining the gradient directions, we rotate each subsequent image with 6 additional degrees in an anti-clockwise manner. The results were uploaded to a contrast map and printed out. Here are the detailed codes and the contrast map. We then add noises to the images by the root sum square model. We extract the B-log image with B value equals to 5, and the noise level is calculated based on 1% of the maximum intensity of the B-log image. The noise level chosen here is 1%. Here are the noise map generated and the data added with the noise. We then perform quadratum Fourier transform with the FFT and FFT shift function. The data points ranged from 0.6 to 0.8 and 1.3 to 1.5 are displayed. Now is the time to generate the mask and apply it. We first generate 4 sectors and a circle in the middle, and we combine the 5 segments together. We rotate the mask 6 degrees each time to generate all masks for all images. We then apply the mask onto the images, and the data points outside of the mask are normalized to mean k-space. Finally, we performed quadratum inverse Fourier transform to obtain the processed data. Now we come to the discussion and conclusion part. After the performance of the experiments, we obtained a colored FA map, which provides a clear view of diffusion directions of fluids inside the brain. We also obtained the noised DWI images, which provide more accurate information and images with high resolution. There are some decisions made that affect the approach of the study. The first one is that we changed the planning of using the FSL toolbox to calculate the tensor for the images, because initially it did not work. MATLAB was used instead. Furthermore, the original result of visualizing data points of range 0.8 to 1 and 1 to 1.2 is not significant. So we changed the range to make the results to be more significant. The problems that we encountered include partially denoised images at high noise level, and the code arrangements can be improved because they are quite chaotic right now. The plan is to use deep learning models to improve the accuracy of noise reduction and to refine the mask creation process to adapt to different datasets. We are also planning to delete insignificant lines in the code. The results now can be applied into clinical settings for visualizing microstructure of the brain and investigate whether there are abnormalities or blockages in the fluid flow of the brain. Also, the denoising methods can be added to current MRI denoising systems. This is the end of my presentation. Thank you.